I think we can zip through that rather efficiently. Then we'll look at connective tissues and then come back and talk about, look at the skin in a little more detail. So with that in mind, I think you need, if you don't already know, you need to know the 4 types of tissue. I've never really been very inspired or compelled by this classification system. But they're listed at the top there, and you just need to know them. Epithelial, connective tissue, muscle, and nervous. So basically, if a tissue, if a region of the body is not muscle, is not nervous, and is not epithelium, we just throw it in that catch-all category of connective tissue. And so we'll talk more about how connective tissue is identified, described, and so on in just a moment. There are a number of different types of tissues. Of course, there are over 200 different types of cells in the body, but in addition, a large number of tissues, and there are gradations between the two, so I wouldn't worry too much about the scholastic distinctions between the various types of tissues. In general, epithelium is that type of tissue that lines the lumens of tracts that are connected to the exterior of the body. So we have the various types of epithelium. For example, the GI tract is lined with epithelium, the respiratory tract, the genitourinary tract. In addition, epithelium can also line ducts, the interior ducts that are not actually connected to the interior of the body. By contrast, connective tissue is all over the place, as we've indicated. Everything from cartilage, bone, blood are all connective tissue. We either, in general, we say that connective tissue is either binding or supportive. I've never found that very helpful either. But traditionally, connective tissue is either binding or supportive, and we'll see how that falls out a little later. You know that the science, at least the traditional division of biological or life sciences devoted to the study of tissues, is called histology. The various regions of the body, we've already encountered a few of these, tend to be sequestered or compartmentalized in a variety of ways. Often these membrane systems separate the compartments both anatomically and functionally. There are epithelial tissue membranes, which basically, again, these are the epithelial cells that we generally describe as a mucosa that rest upon a basement membrane, and then that basement membrane is usually resting on some other type of connective tissue, such as a connective tissue membrane. And these connective tissue membranes can, for example, perform a variety of functions, everything from surrounding organs to providing the serosa and connective tissues that bind, for example, the GI tract or the respiratory tract to the surrounding thoracic or abdominal wall. In the illustration, you can see references to some terms you should be familiar with over here. I don't know how clear these are. Mucous membrane. That is generally a type of epithelial membrane. In general, when we talk about a mucous membrane, we're talking about mucosa. So we're talking about usually a tubule or system that opens to the outside of the body that is lined with epithelial cells and some other structures we'll talk about later, lamina propria and basement membrane. But all those together constitute the mucosa. So these mucous membranes are characterized by a laminal or stratified set of cells that form the mucosa. Serous membranes, on the other hand, are a little more difficult to describe. Serous membranes are basically those membranes that are inside, away from any exterior opening of the body. Pleural membranes would have a serosa. The mesentery of the GI tract is going to have a serosa. So this is usually just sort of a oily, sort of fluid region, membranous region that allows for motility and movement, lubrication. But in any case, the serous membranes do not, as a rule, open to the exterior of the body, such as the peritoneal mucosa, peritoneal membranes and the pleural membranes. The cutaneous membrane is just the integument, the skin. We'll talk a little bit about that. And then you also have the synovial membranes that, for example, line the articulatory surfaces of the cartilaginous joints. I'm sorry, the synovial joints, not the cartilaginous joints. So, as I said, we want to look first at epithelia. This is one of the four basic tissue types. Epithelia, first and foremost, lines those luminal systems that open to the exterior of the body, and it also lines ducts such as the ducts that extend from the salivary glands or endocrine glands. These are also characterized by the presence of epithelia. They perform a variety of functions. The epithelium, in general, as we indicated a moment ago, can be anything from a very flat cell to a very tall, elongated cell. In either case, well, not in either case, but in most cases, those cells will rest upon some type of connective tissue membrane, in the same way that the carpet here might be that basement membrane. Now, so, in general, not always, but in general, epithelia rests upon a basement membrane. The basement membrane sort of prevents blood vessels and various other structures from passing from the interior to that epithelium. So that basement membrane, in some cases, can be very limiting. One of the key characteristics of epithelia is that it's not generally vascularized. We don't have a lot of blood vessels making their way through that basement membrane. Now, we'll talk more about this term later, but many of the other tissue types, such as cartilage and bone, as we've already seen, have cells that are positioned at intervals within a much larger matrix, extracellular matrix. Well, epithelium doesn't usually have much, if any, matrix. So epithelium is characterized by the absence of any type of extracellular or intercellular matrix, some sort of material between the cells that's just generally missing. And by contrast, in fact, almost in contradistinction to other types of tissues, epithelial cells are characterized by the presence of distinct and robust cell junctions. They lack matrix, but the cells themselves are frequently tied together by a number of cell junctions we'll talk about. We want to pay attention to the organization of the cell junctions. Oftentimes, in a number of pathologies you'll be dealing with, it's a failure or problem with those cell junctions that may be the responsible agent for the pathology. So as I say, we'll come back and talk about the cell junctions, characterize those a little bit more. We've already seen in the physiology class, we've talked about transepithelial transport. And so you already know that epithelium is characterized by, in very general terms, an apical surface and a basolateral surface. And we'll see the structures of the epithelial cell that sort of delimit those two regions. But remember, the apical surface generally faces the lumen of whatever tube that epithelium is lining. Basement membrane tends to be more apposed, APOSE, to the blood supply, the ISF in the blood supply. So we've talked about the fact that the epithelium tends to be avascular, but because this tissue is avascular, because it tends to be in a relatively exposed region of your anatomy, wherever it is, whether it's the integument, whether it's the respiratory tract or the GI tract, it takes heavy losses on a regular basis. If we had the means in here to do it, I can promise you it's under you right now would be a little pile of dead skin cells. And it wouldn't be very difficult for an animal, a bloodhound or whatever, to pick up on that. Likewise, you're sloughing off, you're losing just millions and billions of GI tract epithelial cells on a regular basis, and you can probably imagine where those end up. But you get the idea. So for that, if that's true, the epithelial tissues tend to be actively mitotic. We say they are very rapidly regenerative, or actively mitotic. They're undergoing rapid turnover. I made reference to the cell junctions a moment ago. Let's see if, let's go to this illustration first, because you've seen this, something like this before. So down here, the yellow cells would be the epithelial cells, which is in the illustration on the bottom here, which side is apical toward, well, it says it right there. I can't even ask you a question. If I wake up, I could see that. So the apical side is to the top, the basolateral side is to the bottom, as you can see here. And so when we talk about different types of cell junctions, what we're going to find, it's not terribly well indicated here, but oftentimes there will be an array of cell junctions that bind these two epithelial cells together, again, depending upon the cell type, its location, and a variety of other factors. Up here, they're making reference to a specific type of cell junction we'll look at in more detail in just a second, called a tight junction. And those tight junctions, as you can see, tend to be clustered toward the apical surface. And they effectively prevent, I don't think it's in the slides anywhere, so you may want to write this down. They effectively prevent paracellular movement of substances between the epithelial cells. Paracellular, we're not talking about taking a parachute out on the beach. Oh, if you'd like, we can. I think that might be on a Friday, if I can have more appropriate. But this is paracellular, and in the case of paracellular, we're talking about movement of something between the epithelial cells, and in general, that's not desirable. Let's say you had sausage for breakfast this morning, this is the line. You could imagine this to be the lining of your GI tract, the lining of your intestines. Would you want that sausage to slip into here and get into your blood without passing through your GI tract epithelial cells? What would be the problem with that? Any of you have sausage in your blood right now? And you knew, because I think you could probably list some of the causes. What would be the primary problem associated with blood sausage? What would that immediately incite?
Clotting?
Right, so, well, okay, that's probably true. Although that might help me, because if I can get rid of some of the GI tract uptake, I'm probably going to be better off. But what else would probably happen? Yeah, I think you're right. I think that's a foreign substance in your blood, right? Well, probably not in my blood at this point, since I eat sausage on a regular basis. I'm turning into a breakfast sausage, in fact. But in general, you don't want foreign proteins in your blood. That's pretty much an alien substance, a foreign substance. And the immune system would probably have something to say about that. You might get a severe immune response, immunogenic response. So yeah, we can go on and on about it, but I just want to make sure you have a general understanding of why these tight junctions are so important. And in certain types of pathologies you learn about in your clinical coursework, they don't do their job so well, and that can be a problem. So there are only a few types of substances. Water, for example, for reasons we won't get into now, tends to have the ability to pass to some extent across in a paracellular fashion. But in general, most everything that's taken into the blood from the intestines should pass through the epithelial cells. And that, I guess we would say that the tight junctions are very important in making sure that happens. By the way, if you remember that the dentist will often talk about occlusion and malocclusion. What do they mean by that? You're right, the alignment, the idea that the teeth are properly aligned and making sure that they're coming together properly. This is sometimes called the zonula occludens, because again, these two cells are meshing together properly to prevent paracellular migration. And we'll see that in one of these illustrations up here. You can see, so as we look at the graphic here on the upper left, that's an illustration. It's kind of like a series of rivets, if you will, in the adjacent epithelial cells. And so those are membrane proteins, transmembrane proteins, and their function there is to sort of rivet those two cell membranes together to ensure that nothing passes between them. Oops. Now, typically, let's go back to this illustration again. As I say, typically, in addition to the tight junction, the tight junctions are basically like, I don't know, what's a good example? Like aluminum doors, very inexpensive aluminum doors, and they have a nice hermetic seal, but they're not very robust. Let's say we can break through them. They're not very strong. By contrast, deeper along the epithelial cell, I guess that's a relative term, but deep to the tight junctions relative to the lumen, you typically have additional cell junctions that are much more robust. And there are two general types. They're all called anchoring junctions. Anchoring junctions, I don't know if that's in the slide or not. Yeah, there we go, anchoring junctions. And there are usually two types. Get a little bit confusing. There are adherens junctions. Adherens junctions, and if I see that right down here, adherens junctions, and desmosomes. Those are the two primary classes of anchoring junction that provide the load deep to the tight junction. And you can see several illustrations. This would be a desmosome here on the left. This would be an adherent junction on the right. And they're very similar. They differ in a number of ways, but for our purposes, they differ in two primary ways. Number one, position. In general, desmosomes tend to be closer to the basement membrane. Desmosomes tend to be closer to the basement membrane. The desmosomes tend, both adherens junctions and desmosomes aggregate. They come together in clusters, but the adherens junctions clusters tend to be larger. We just call the desmosome clusters plaques. So when you read about epithelial plaques, oftentimes the reference is to those desmosomes. But the second major difference between the two that you want to pay attention to is, if you'll notice in the illustration, here's the cell membrane on either side. Here's the cell junction, but you'll note that the cell junction in both cases is attached. It isn't so clear here, but it's attached to the cytoskeleton of each cell. All right. Desmosomes attach to the intermediate filaments. There are three major types of cytoskeletal filament in most cells. There are microtubules, microfilaments, also called actin filaments. Microtubules are big, they're thick. The microfilaments or actin filaments are very small. And then in between those two, there's a third filament. It's called an intermediate filament. Isn't that convenient? It's called an intermediate filament. And so desmosomes, and you can see them here, these are spaghetti doodles sticking from either side. Those are the intermediate filaments. So in effect, you've got the cytoskeleton of one cell bound to the cytoskeleton of the other cell. So that can be a very robust joint. That's a desmosome. So an adherens junction does something similar, except it doesn't interconnect intermediate filaments of adjacent epithelial cells. It interconnects adjacent microfilaments. It just joins microfilaments together. So otherwise, they're very, very similar. What you'll notice then is you have the cell junction here in this particular desmosome. You have a region, in this case, it's sometimes part of this plaque, this large plaque that you see where these things collect together. But here's the important thing to pay attention to. These are the interlocking junction proteins, And in some illustrations, you'll see them extending all the way across the cell from one side to the other. They don't usually do that. These CAMs, they're cell adhesion molecules. usually interlocked when they extend from either side of the cell junction, or either side of each cell membrane. And when they bond together, they link together between the cells. The most common, and it's listed there on the slide, the most common cell adhesion molecule is something called a cadherin. But there are a number of these, and we're discovering more of these in various tissue types. Cadherins are the most common type of interlinking protein in a desmosome, or for that matter, in the cell adhesion, in a cell junction generally. Now, the only other type of junction mentioned here in a couple of places is the one at the bottom here. This is called a hemidesmosome. Hemidesmosome. And these are different from the desmosomes in that they're not linking two cell membranes together. They're basically binding the basolateral surface of an epithelial cell to the basement membrane, binding it to what's called a basal layer. So it's only half a desmosome, right? That's the logic there. Otherwise, the hemidesmosome is still attached to the intercellular keratin filaments or intermediate filaments. In the skin, the intermediate filaments are keratin filaments. That's what gives your nails and hair and your skin the strength that it has or doesn't have. So, yeah, the next few slides here just talk about the desmosomes, and you have another illustration here. So, and you look at this, I think you can imagine, so if there is a problem with cell-cell linkage, epithelial cell junctions, oftentimes it's a deficiency or problem with the CAMs. There are, it's not usually compatible with survival, there are certainly some genetic mutations in which the protein is not expressed properly, and that can cause all kinds of problems. Can you think of a disease in which your cells detach from their location and go somewhere else, perhaps to take up a home? Yeah, that's the metastasis of cancer. And so there's another type of cell junction called a catenin. We won't be C-A-T-E-N-I-N. And it's fairly clear that certain, cancer is characterized by a number of things that have to happen, things that have to go wrong before you really have a problem. But certainly in most cancers, there is this problem with metastasis, and oftentimes, not always, oftentimes it's associated with a failure or breakdown of these cell junctions, such as the catenins, to do their job. That enables the cell to migrate and do some other things. Yeah.
So that would be like one of the mutations in cancer that would lead to metastasis. Is that what you're saying?
Well, I guess I would differentiate or dissociate those two things. What I was saying initially is that there are some children, for example, born with genetic defects in which there's improper, inadequate expression of cell adhesion molecules, and they have all kinds of problems. There are a number of proteins involved here, and depending upon the degree of underexpression or inappropriate expression, that can lead to problems. Cancer is a little bit more complex, and I wouldn't want to overgeneralize. What I'm suggesting is that in many types of cancer, many types which originate in epithelium of one type or another, in order for, because cancer involves proliferation, abnormalities of various types, but in order for that metastasis to occur, yes, there must be some aberration in the cell adhesion, in the cell junctions. So we've talked about tight junctions, we've talked about desmosomes, we've talked about adherens junctions. There's yet another type of junction that we should mention in this connection. It's a bit of a cell junction. I'm not sure, I'd probably cross out anchoring up there, because this is not really an anchoring cell junction. The gap junctions serve a different purpose. The gap junctions are present in a variety of tissues, for example, cardiac muscle, certain types of smooth muscle. Most textbooks will tell you that they're not present in the brain. Recently, we've even found gap junctions in cells of the brain. And what they are, very simply, are passageways, openings, that allow the cytoplasm of one cell to pass directly into the cytoplasm of an adjacent cell. They're just little apertures. And they go by a variety of names, and I guess because you'll see them, you see all these terms, you should know them. Gap junctions are also called electrical junctions, because if there is a depolarization, and you now know what a depolarization is, if a cell depolarizes and it has gap junctions in its membrane, that can be a mechanism whereby an adjacent cell can also be rapidly depolarized. This happens in the cardiac muscle of the heart, for example. These are also called connexons. Pay attention to the spelling of this. So there are three general terms: gap junctions, electrical junctions, and connexons. The term connexon refers to this whole complex. Connexon, O-N. But wonderfully for the people who named this thing, connexons are made up of individual proteinaceous connexins. So this would be a connexin, and so on. So INS, multiple connexins make up one connexon, otherwise known as a gap junction or electrical synapse. I'm sorry, electrical junction or electrical synapse, same thing. What's the difference between a gap junction and an ion channel? What's the difference between a gap junction and an ion channel? Yeah.
Aren't the junctions between different cells, and channels are only for particular cells?
Well, I agree with you in the first part, but I'm not sure what you mean exactly. So an ion channel is a channel between what? All right, the extracellular environment, and I think that's the extracellular environment in the interior of the cell. So that's different. A gap junction is directly between two adjacent cells. It's like these motels here, you just go right into the adjacent room. Not that I'm familiar with that process at all. So any questions about cell junctions? I think that, again, you'll see why it's worth knowing a little later in your clinical coursework. But let's talk a little bit about classification of epithelia, since that's what we're looking at now. Classification of epithelia is quite trivial, but you do need to know it because it's just relevant in the literature. Basically, there are three general shapes, three general type or shape classifications of epithelial cells: squamous, cuboidal, and columnar. Squamous, flat, sort of flat shape, cuboidal, sort of squarish shape, and then you can see the elongated shape of the columnar epithelium. Likewise, or similarly, we say that epithelia is either simple or stratified. If it's simple, there's only one layer. If it's stratified, the epithelial cells are stacked. Now, some of these are much more common. You see all the possible variations here. It's often the case that squamous epithelium, for example, can be simple, or it can be stratified. Both are very common. We'll look at examples of that. With respect to cuboidal epithelium, simple cuboidal is much more common than stratified. Basically, you only find stratified in a few sparse locations we'll talk about. But simple cuboidal epithelium is quite common. Similarly, simple columnar epithelium is very common. Stratified columnar epithelium, in my opinion, is misnamed, but we'll talk about that in just a moment. But you will rarely encounter this, and it really won't even come up in your clinical studies. Now, there are a couple of epithelia that really don't fit into any of these categories. One is called pseudostratified, and it gets its name from the fact that as you look at it in a microscope, it appears, and this isn't a particularly good illustration, it appears to have multiple layers, but that's only because the apical surface may start out here, but the basement surface may end up over there. Right? So when these cells are interdigitating and squirming, they often have the appearance of having multiple layers in a microscope slice. The other one is a transitional epithelium that we see in the bladder, and it's called transitional because its shape, its appearance changes depending upon whether the tissue is distended or relaxed. So there are two here that don't fit into the traditional scheme that you see over on the left. The pseudostratified, usually it's ciliated pseudostratified, and the transitional. Yes?
Would the transitional epithelium be found in the stomach as well?
No, the only place you're going to encounter is in the bladder, urinary bladder, and the ureter. Does that make sense? This morning I noticed when I was looking at this table, I think I got this table from the Open University text. I think that's where this came from, but I saw a little error up there. But this chart, whatever this is, the little table on the right here, takes you through the various types. It talks about the simple squamous, the simple cuboidal, and so on. And then it gives you the typical location. I do want you to know a few of these. We'll talk about that in just a moment. And then it describes the function, and I want to talk about that over here too, because the function of the epithelium depends very much upon its shape. But up there I noticed that it said simple columnar epithelium, so just maybe correct this, because every year some student finds this and I only happened to see it this morning. Simple columnar epithelium. Simple columnar epithelium is not generally ciliated. There are a few exceptions. We'll mention those later. Most simple columnar epithelium, such as the lining of the intestine, has microvilli, not cilia, microvilli. It's also called brushwood. So we can classify the epithelium not only by the shape, as I've indicated, but by function, and I'm going to talk a little bit about this. So epithelia can be protective. That would be one functional or physiological classification of epithelium, independent of its anatomical classification and physiological classification. Epithelium can be specialized for exchange. What do we mean by exchange, do you think? Yeah, exactly right. Gases or whatever passing through it. So you might be thinking about which of those shapes would be best suited for that. Some epithelia is basically designed for shipping and receiving. So if you're going to be an epithelial cell that's going to get in the shipping and receiving business, probably you're going to need a few more raw materials, more space, more room, more activity than would be afforded, for example, by an exchange epithelium. And we're going to use the term distension. Transitional epithelium is specialized for distension and relaxation. A lot of epithelia cannot handle that type of mechanical stress, but the transitional epithelium can. And then, as we've indicated, some epithelia have cilia or microvilli. In a couple of cases, one very important case that you'll learn about later, it has both. The cell has both cilia and microvilli, most acute. But of course, when we talk about cilia, let's say we have a ciliated cell, a ciliated cell, what are the cilia doing? What's their function? Yes, well, the cilia, microvilli are more important for surface area. Cilia basically are little hairs that on the apical surface, and they're doing this all day. And as they're doing that, they're moving something along. They're passing something along. So this would be very important, for example, on the upper respiratory tract. So here I'm going to talk a little bit about the functions here. So the simple squamous epithelium would be well suited, for example, to exchange of gases. There's not much to it. It's flat. Gases can pass readily right through it. The simple cuboidal epithelium, you will find, is mostly associated with secretion. It's a lot of glands, a lot of exocrine glands, and even some endocrine glands are characterized by the cuboidal shape. Simple columnar, again, a lot of real estate here. This is shipping and receiving, even more so than the secretory cells. These cells often have a lot of things going on simultaneously. And in fact, in the GI tract, where you find these so abundantly in the intestinal lining, they're not just doing shipping and receiving. They have whenever they take something in, they process it. They have a lot of enzymes in there that work on it and dispatch it and reformulate it and so on. And likewise, they have to modify the substances that they discharge as secretory products. So very, very busy, lots of cytoplasm. So these are very large cells for that reason. Simple columnar, again, typically along the GI tract and portions of the reproductive tract too. Ciliated, the most common example that you will encounter is the ciliated pseudostratified columnar epithelium. Ciliated pseudostratified columnar epithelium that is characteristic of, for example, the upper respiratory tract. Also lines portions of the reproductive tracts. And once again, when you see the term cilia, 98% of the time that epithelium is doing this and pushing something along. And in the respiratory tract, it's called the mucus escalator, for example, and we'll talk about that later. And what has only been discovered very strangely in the past, I would say this discovery has only emerged in the past 10 years. Maybe somebody can research this, perhaps even more recently than that. Did you know that virtually all of the cells of your body, 90% or so of mammalian cells, have a single, at least one, but always one single little cilium? And what does it do? We don't know, but we know if it's not there, the cell has a lot of problems. And so that's an active area of research. But it's usually called the primary cilium, and it's thought to be involved in cell communication. But what is quite clear is that if for any reason it's not present, that tissue does not thrive. And so we talked about the pseudostratified columnar. Most commonly, it is ciliated. This, as we've indicated, is most abundant in the respiratory tract, and again in a few other locations, such as the reproductive tracts. Very common, especially in simple columnar epithelia, are these cells. It looks a bit like a paramecium here, but this is a goblet cell, and you can see this is a columnar epithelium here, and there'd be a goblet cell there, there'd be a goblet cell there. These are, for the most part, mucus-secreting cells, And you'll notice they often, depending on the location, have microvilli as well. They may or may not, depending on the colon, or depending on the location. For example, in the colon, they don't have an abundance of these microvilli. What's the difference in those two words? Which do you prefer? They're two different words. It's kind of like nauseous and nauseated. Does this class make you nauseous or nauseated? I see, I'm both, frankly. I'm both nauseous and nauseated, frankly. Shut up, I'm going to get worse, and it's going to be unpleasant in here. So the first is a noun. So mucus is the slime. This is the adjective. So properly, you don't always see this observed, this would be a mucous membrane, and a mucous membrane may have mucus on it. Supposedly, you are nauseated because I'm nauseous. Nauseated is what happens to you. The reason that you become nauseated is because something else is nauseous. It causes the nausea. So. I think this is just a summary of what we've already talked about. We've already talked about squamous epithelium. So let's just quickly review some of the points. Simple squamous is important for exchange. We'll look at a couple examples of this in just a moment. Simple squamous would be found wherever gas exchange and exchange of nutrients must be optimized. By contrast, stratified squamous, this is the tissue, this is the epithelium that goes into battle. This is a protective epithelium that suffers heavy losses. And so we talked about the fact that these cells need to be rapidly replenished. Your integument consists of, for the most part, stratified squamous epithelium, except in a few locations. Cuboidal, both stratification is rare. We'll look at an illustration in just a moment. But in general, cuboidal and columnar epithelium, high secretory or absorptive activity. A lot of things going on. These cells have a lot of metabolism. Transitional epithelium is characteristic of the urinary bladder, and it's designed to withstand the mechanical forces imposed upon that. Again, not suggesting for a moment that any of you need to go to the bathroom. So here's an example of simple squamous epithelium. What are we looking at on the left? Anybody recognize this? Boy, you're going to recognize this by the end of the course. This is, going to give you a clue. Huh? Well, that's the alveolus. See, yeah, I think I see. Renal corpuscle, remember the kidney? So this is the Bowman's or renal corpuscle. And remember, you probably remember this now as you start to, as the shake set in, you had to go through this all before. There are a couple of membranes associated with this structure. This is the parietal layer of the capsule. This is the mucosal or interior layer. But both of these are going to be simple squamous epithelium. But this one, you can see how it's very flat. Alveolus over here. This is called the type 1 pneumocyte. Type 1 pneumocyte, very flat. Nucleus over there, optimizing exchange. And of course, it's very clear here, but if you look right there, probably what we're looking at is down the barrel of some blood vessels, because of course gas exchange in the lungs is between the pulmonary capillaries and the interior spaces of the alveolus. This is stratified squamous. This is in the lip. Now the lip is a little bit funny because it is a transitional region in that the skin out here, if you reach over and scratch the person beside you very forcefully with your fingernail, you'll notice that, I don't have my skin left anymore, but a little white line will appear. Right? And that's because that stratified squamous skin in the integument is keratinized. That's mostly just the dead stuff up there. But in the lip, do it to the classmate beside you. You'll find it's a little different. It's much less keratinized, and then you go into the interior of the mouth, it ceases to be keratinized. So the lip is actually a transitional area. You have both keratinized, dead husks of keratin. What was keratin? We talked about it before. Intermediate filaments. Yeah, we're going to see that it's basically just those intermediate filaments that are extruded as these cells sort of emerge from their superficial origin to the exterior of the skin. They progressively die, and as they do so, all that's pretty much left is that keratinaceous husk. When you eat corn on the cob, have you ever noticed the product that results from the digestive system? Somebody? All right. But in any case, somehow I don't know why I got off on that. But anyway, the keratin is what's left, right? So it's basically, but in the lip, and we're sort of looking at this transitional region, we don't see much of that keratin because of that transition. By contrast, here, this would be the integument, the skin. We're going to look at this a little later. The skin has both a dermis and epidermis. This is the dermis down here. This is the epidermis here. These little protrusions are what are called dermal papillae. And what we'll find, as we discuss this in a little more detail later, these epithelial cells here, they originate down here, and then they migrate out toward the superficial regions of the skin. But as they do so, you'll notice out here, you don't see many cells, right? You don't see many. So all that's really left is keratin. And so all of this out here are just keratin husks, kind of like the corn husks that you see as a product of your digestive system. Now this, please don't cry in terror as you look at this, but this is the cornea of the eye, not suggesting for a moment that your cornea is purple. This has been stained, but you'll notice this is a stratified squamous epithelium. It's fairly tough. Your eye is at fairly high pressure and has to be fairly robust for that reason. But this is the stratified squamous epithelium. By the way, I didn't state this, but I should have written it down. I noticed it wasn't in the notes. All of these epithelia, I should say, all of the stratified epithelia are named for the most superficial layer. All of these stratified epithelia are named or designated for the most superficial layer. And that's important because as you look at this, how would you describe these cells? Yeah, whatever you say, it's clearly not squamous. So that's why, and that's generally the case. The cells at the interior are not necessarily the same shape as the one. So they just by rule say that whatever is on the outside or the superficial is going to be used to name the epithelium. Here we have a simple cuboidal. This is in the kidneys, for example, renal tubules, simple cuboidal here. And then a stratified cuboidal, where you normally find these over here, it talks about the male urethra. The most common location for this stratified cuboidal is in the ducts of glands, salivary glands, certain exocrine glands that go to the GI tract. This is about the only place you're going to encounter stratified cuboidal. And sometimes you don't even observe it there. We have several illustrations of these. So this is not a histology course, but if I were to provide you with an image such as this on the test and point to those cells, I would expect that you'd be able to give me a morphological classification, simply stating that they are, what you're basically asking yourself, is there one layer of these cells or more than one layer, and what shape are they? So these would be cuboidal. I would not ask you to identify stratified cuboidals, so don't worry about that. You wouldn't have to identify that. This is one, and we'll stop there and take a break, but this is one you would definitely have to be able to identify, because this is going to be very important. What we're looking at here is a villus of the intestines. This is a villus here, finger-like projection. We can see the simple columnar epithelium with the microvilli, also called the brush border. This is a region called the lamina propria. Lamina propria is characteristic of this type of tissue. And then, of course, this is your breakfast sausage. Right? This is the lumen of the intestine, and of course, it's these cells that are going to go to work on all that. All right, let's take a break.